1. The next evolutionary trend that we see in animals is transitioning from having no fluid-filled body cavities, being acoelomate, to having a false fluid-filled body cavity called a pseudocoelomate, or having a completely mesoderm-lined fluid-filled cavity called a coelom in eucoelomates. So acoelomates are typically very small and very flat organisms because they have to be able to nourish all of their cells and get oxygen to all of their cells. In order to do that, it's very difficult to do without any spaces inside of the body to move those resources around. So flatworms, for example, are a good example of acoelomates. And you can see with these colors, red representing cells derived from ectoderm, blue or purple representing cells derived from mesoderm, and yellow representing cells derived from endoderm. Okay, so in acoelomates, all three germ layers and the cells derived from those germ layers are mushed together in the adult. Now, in pseudocoelomates, like this roundworm here, you can see that there's a space in here, but it doesn't line the cells derived from the endoderm. It doesn't line the gut and the other organs. So there is this cavity, but not being lined with cells derived from mesoderm makes it a pseudocoelomate. In eucoelomate organisms, which include things like earthworms, annelid worms, and you and I, we are eucoelomate, you can see that the mesoderm in the adult organism lines all of the organs. So you can see the gut here is completely lined with mesoderm, and this enables eucoelomate organisms to have more complex organs because the cells and the tissues that comprise those organs are held in place by this substance called peritoneum, which is derived from mesodermal tissue. All right, so in looking at a tree and phylogeny of all animals, we can see certain trends that are reflected within it. So the Porifera are considered by many zoologists to be very basal animals. They have a lot of ancestral characteristics, as we've just talked about, having no planes of symmetry, having only a cellular level of organization. And then when we get into radially symmetric organisms like comb jellies and cnidarians, true jellies, and other organisms, we see a tissue level of organization. And then it isn't until we get into organisms with bilateral symmetry that we find organs and organ systems being found, and as well as being triploblastic or having 3 germ layers. So these things seem to go together: bilateral symmetry and the presence of 3 germ layers. Okay, so we're going to look at all of these groups: the Porifera, ctenophores, cnidarians, acoelomate organisms, deuterostomes, Lophotrochozoa, and ecdysozoa. So starting with the phylum Porifera, these are the sponges, and by many accounts they are among the most ancestral types of animals. They only have a cellular level of organization. They have relatively few different types of cells that comprise the adult organism. So what are those cells, and what are those structures that comprise a sponge? Well, within the sponge, let's start with these choanocytes over here, these collar cells. The choanocyte, you might look at that and say, hey, that looks kind of familiar. I feel like we've seen something like that before, and we have. In the choanoflagellata. So that choano prefix, which means collar, has come back from the choanoflagellata to the choanocytes that are present in sponges. And like Choanoflagellata, they have a single flagellum that is surrounded by this collar of finger-like projections called microvilli, and the flagellum creates this tiny little current that brings food particles through the collar, and they get trapped in the mucus. And then once they get trapped in the mucus, they can be brought into the cell through vacuoles, and those vacuoles can be transferred to other types of cells, like for example these amoebocytes, that can move those nutritional resources where they're needed in the organism. So here we can see some choanocytes that are lining the inner cavity, or the spongiocele of this particular sponge, which is an asconoid type sponge. How does the food get to these choanocytes, and how do they use those flagella to pull water through? They have pores. Porifera means bearing pores. They're full of holes. And the pores in an asconoid sponge are made up of special pore-producing cells called porocytes, shown here in this kind of lavender color. And in asconoid sponges, the openings for water entry, called ostia, well, you don't see the ostia on here, are made up of individual porocytes. So the porocytes bring water from the outside into the spongiocele, where the food particles can be consumed by the choanocytes. Out on the outside, there is something like an epidermis in the sponge, and that is made up of another type of cell called pinacocytes. And pinacocytes can help give structure to the outside of the sponge. All right, so water is flowing from the ostia into the spongiocele and out of this way for water to exit, which is called the osculum. The osculum is the hole for water exit. Okay, one other thing to point out to you are these spicules, which are typically made of some type of mineral-like substance, either made from silica or silicon dioxide or calcium carbonate. So sponges can be either calcareous or siliceous, or sometimes they're neither. Sometimes they just have protein fibers of a protein called spongin that makes up the body of the sponge. And these layers between the epidermis and the lining of the spongiocele, that's called the mesohyl. That's what this arrow is pointing to here. Is this where you find the amoebocytes and the spicules, and before you get into this chamber here, that is the mesohyl, which is kind of like a matrix for the amoebocytes to move around and the spicules to be formed. All right, we've got three sort of generic terms for the body plans of sponges. And these are not taxonomic groupings. It's just a description of the way that the sponges are constructed. And they go from the simplest to the most complex. So ascon sponges, or asconoid sponges, are the simplest. This is an ascon sponge. Syconoid sponges are a bit more complicated. And imagine we took a bunch of these ascon sponges and sort of stacked them up on top of each other. That's what we'd be seeing here with this syconoid sponge. And then leuconoid sponges are even a bit more complicated than either the asconoid or the syconoid sponges. So the leuconoid are the most complex type of sponge. So we can remember ASL is the order from the simplest to the most complex body plan in sponges.